Okay, so the next topic at hand is gene expression. Again, you may be familiar with this from your Gen Bio. You should be, actually. But there is a additional component that you may not have heard about in Gen Bio, and that has to do with bacteria. So they pretty much do the same thing with respect to transcription and translation, but with respect to their gene structure and other things, they're a little bit different too. So we'll learn it. We'll go through it. So this is gene expression. One of the things you will notice as a main difference between eukaryotes and prokaryotes, just like cellular respiration, is where things happen. Location of things. Now, because bacteria do not have compartments, they don't have a nucleus, they don't have Golgi apparatus, they don't have anything that is membrane bound, like a membrane bound organelle. Everything happens in the cytoplasm with respect to gene expression. So transcription happens in the cytoplasm as well as translation happens in the cytoplasm, just like you see here. This part here is transcription, and this part here is translation. The interesting thing about bacteria, and it's in the picture, and you might overlook it. You may look at the picture and say, oh, that's so cute. But then you don't realize that it's actually telling you something. This here, look at that. It's transcribing. At the same time, it's translating. Now, that cannot happen in our cells. But in bacteria, look at it, it's still transcribing. It's not done transcribing yet. But the beginning of the transcript, it's already translating into protein. That is called co-transcriptional translation.
It's doing it simultaneously.
Yeah. Co-transcriptional translation. What does that mean? At the same time I'm transcribing, I am translating. Now that can happen in bacteria, for sure. It cannot happen in eukaryotes. No way. Why? Because in eukaryotes, transcription happens in one location, the nucleus, and translation happens in a different location, the cytoplasm. So there's no way they can do it at the same time. You have to finish transcription completely for eukaryotes. Then the RNA leaves the nucleus, but by then it's completely done, completely made. Then it binds to the ribosome for translation. So, very important difference. In bacteria, there is co-transcriptional translation because they lack compartments. There's no nucleus. So they are able to do that. In eukaryotes, it's different. You have to finish transcription first before you start translation because they happen in two different compartments. All right, so that's one key difference. The other thing is that gene expression. You've heard about gene expression in Gen Bio, and somebody told you probably that gene expression is the same as the central dogma. I mean, listen to this word, central dogma. Sounds very important. It is. Without this, your cell cannot survive. The main purpose of gene expression is to make RNA that is translated into protein. Most of your genes will do this. Your DNA is copied into RNA. So I stress that word because some students think their DNA changes to RNA. If that happens, that will only happen once because you'll be dead after that. You don't want your DNA to change into anything. Trust me. Your DNA is copied into RNA, and it's not the entire DNA. With respect to gene expression, you're only copying the gene that you want to express. For example, the catalase gene. You don't have to copy your entire DNA just to get the catalase protein. All you have to do is copy the catalase gene. That is called transcription, coming from the word to transcribe. So remember, it's copying the DNA, not changing the DNA. Big difference between the two. One you will survive, the other one you will not. So now I have a copy of the gene in the form of mRNA. Does anyone know what the little M stands for?
Messenger.
Messenger. Yeah. So let's keep going with the catalase example. Now I have the message to make the catalase protein. And that is called translation. And who's involved in that? The ribosome, the transfer RNA, and the ribosomal RNA. Sorry, the ribosome, the transfer RNA, and the messenger RNA. The ribosome comes with the ribosomal RNA. The transfer RNA comes with the amino acid. We'll talk about all of that. And the messenger RNA comes with the message. So all three types of RNAs that you see here are involved in translation of this message. You gotta catch this part. The mRNA has the message. The tRNA delivers the amino acids. And the ribosomal RNA is the scaffolding of the ribosome. So all three types of RNAs are involved in translation of the mRNA. But catch this part. But not all three of them are translated into protein.
You said the tRNA delivers amino acids?
Yeah, you'll see that. We'll get into more detail. But you gotta catch this part. All three of them are involved in the translation of mRNA. But not all three are translated. You catching the difference between the two? The tRNA is not translated into protein. There is no such thing as protein tRNA. There's no such thing. Only the mRNA is translated into protein. Obviously, different kinds of proteins, depending on which gene you're translating. The ribosomal RNA, same thing. The ribosomal RNA genes are transcribed, yes, but they're not translated. There's no protein that's called the ribosomal RNA protein. There's no such thing. So be careful of that. What happens is you have genes that code for tRNA, and you have genes that code for ribosomal RNA, but that's all they do is transcription. That's it. From those genes, you only make RNA, the tRNA and the ribosomal RNA. You will never have a ribosome sit on the tRNA to make a protein because it doesn't happen. That's not what they're there for. You'll notice that the tRNA behaves like a delivery truck of amino acids. And when do they serve that function? When the tRNA delivers the amino acid to the ribosome that's translating this mRNA. The ribosome? Yeah, the ribosome. That's the ribosomal RNA. The ribosome is made up of ribosomal RNA and a whole bunch of proteins. So now you know. You gotta pay attention to this part when we start talking about it, that all three types of RNAs are involved in translation. They just do different things. This one has the message, this one carries the amino acids, and this is the scaffolding of the ribosome.
So,
Yeah.
question. If there's a question saying only mRNA is used for translation, it can be wrong, right?
To protein. Only the mRNA is translated into
Yeah,
protein.
correct? Or
It is correct.
But you said it has to be the three of them.
All three of them are involved in the translation of the mRNA.
The mRNA.
But the mRNA itself is the one that's being translated. So that's an example of that would be if I have the knowledge on how to build a shelf, that would be the mRNA, the knowledge, the message. You got it? And then I grab a hammer and a screwdriver. The hammer and the screwdriver, those are my tools. The knowledge with the tools. That's what's gonna build my shelf. You see what I mean? Watch, the knowledge and the tools. It's no different than that. Yeah. So with the knowledge or the message of making catalase, this is the sequence for catalase. Translate it. Well, who's gonna read the sequence? The ribosome, which is made up of ribosomal RNA. And who's gonna bring in the amino acids? Because we're making proteins. The monomers of proteins are amino acids. It's gonna be the delivery trucks that transfer RNA. So these guys end up being more like tools. You're using them as tools in the translation of the mRNA. When it comes to the structure of the gene, eukaryotes versus prokaryotes, it's a little different. In eukaryotes, most, not necessarily all genes, but most genes have what's called exons and introns. I studied something at the Cleveland Clinic that the entire gene was one exon. So I didn't have to worry about splicing and all. I just happened to be lucky that way. So the entire gene was a single exon, so there was no splicing involved. But most genes are like this. They have exon regions and intron regions. Now let's look at what they are. Let's say, for example, this is catalase again. Catalase will be actually made only if you get rid of the introns. Now, you don't want to get rid of the introns on the DNA. Remember I told you, you don't want to mess with your DNA. So don't make that mistake. This is DNA. This is what, for example, the catalase gene would look like on your chromosome. Let's say, for example. And I just told you, to make catalase, you have to get rid of the introns, not on the DNA. Because if you study this a little bit more, you're going to realize something. For catalase, these sequences are useless. But you could have 5 different genes inside one of these introns that have nothing to do with catalase. You don't want to get rid of that intron from your DNA. You don't. So what do? You copy this sequence into RNA. So I tell you, don't touch the DNA. Yeah, here, let me copy it to RNA. I get an exact copy of this as RNA. And then I give that to you, and I say, do whatever you want with this one. You can do whatever you want with that. But don't touch the DNA. You mess with the DNA, you're going to be in trouble for the rest of your life, if you survive it. So this is exactly what happens. What happens is your DNA gets copied into RNA, an exact copy. You'll see that. And then the cellular machinery will realize, oh, that's an intron. In the copy, the RNA, it will realize that's an intron that I need to remove. That's called splicing. So you're going to remove the introns in the RNA, not the DNA. Prokaryotes, bacteria, they don't have introns. That's a major difference. Very important difference for you to know that. They don't have introns, so there is no splicing.
Eukaryote and eukaryote?
Prokaryotes do not have introns. There's no splicing. Eukaryotes do. But we do have some things in common. There's a promoter sequence here, and there's a promoter sequence here. The promoter is like the light switch. You push the button, the light turns on. You push the button, the light turns off. That's exactly how it behaves. The promoter is like the control switch of the gene. It turns on the gene.
The eukaryotes don't have a promoter?
No, they do. Promoter. So there are some things that are in common, but there are some things that are different. And you'll see that as we go through things. So pay attention to the introns being spliced from the RNA, not the DNA. The arrangement of things. This one is crucial for your understanding. Let's say, for example, we have 3 genes involved in the metabolism of a sugar. We tend to be, eukaryotes, tend to be so messy. We would have the first gene on chromosome 3, the second gene on chromosome 8, and the third gene on chromosome 9, whatever. Why? Because we have real estate. Our DNA is 3 billion bases long, and we can afford to be messy. Each gene will have its own promoter. So let me explain it again. In the cell, let's say it's metabolizing a sugar, and there's 3 enzymes involved in metabolizing this sugar. Don't expect the genes for those 3 enzymes in eukaryotes to be right next to each other. They're not. We're not that organized. They're going to be on different chromosomes. And each one of them will have its own promoter to regulate its expression. So 3 genes, gene A, B, and C. A is on chromosome 3, B is on chromosome 8, C is on chromosome 9. But they're all involved in the same metabolic pathway. That's not how bacteria does it. Bacteria, they are so organized because they don't have enough real estate. Compared to our chromosome, the size of our chromosomes, their chromosomes are relatively small. So they can't afford to be messy. So what they do is, watch this. In this case, they're showing 4 genes. The 4 genes involved in the metabolism of, let's say, a sugar, a specific sugar. They will all be organized right next to each other, A, B, C, D. And they will all be controlled with one promoter, right here. This promoter will control the expression of all 4 genes at the same time. Big difference between us and prokaryotes when it comes to how the DNA and the genes are organized on the chromosome. We tend to be a little bit more messy. The genes are on different chromosomes. That means each gene will have its own promoter. But in bacteria, they're more organized. All the genes involved in the metabolism of, let's say, lactose will be right here, yeah? And the promoter element controlling all of them right here. And they're very organized that way. And that's called an operon. That's called an operon, the collection of the control elements. There's another control element called an operator we'll talk about. The collection of the control elements with the structural genes. The genes that are involved in the metabolism of a specific metabolite, like sugar or whatever. Everything is neatly organized in a collection of things called the operon. We don't have operons. Only prokaryotes have operons. So if you find an exam question talking about operons right away, it's prokaryotic. We don't have operons. We tend to be a little bit more messy. Questions on it so far?
You said it's a collection of control elements of what?
For which one?
Operon.
The operon is the controlled elements along with the structural genes, these guys. The genes that are actually doing the metabolism of that sugar. They're all gonna be in a nice, handy cassette kind of thing called an operon. Now there's something else upstream called the regulatory gene. We will talk about that, and this thing will blow your mind. And if it doesn't, you are hard to please. When I was a student, I was like, wow, that's amazing. Wow. This is just bacteria. What do they know? Oh, they know a lot. They know how to do this. You'll see. Hopefully I'll impress you. All right, let's go through transcription. Now what we're talking about here, if you could notice here, it's pretty much the same in prokaryotes and eukaryotes. There's only one enzyme involved in transcription, thank God. Not like DNA replication. And I'm not going to harass you with this one right now, because I know you probably didn't study it yet. But I will harass you 1 week from now. With DNA replication, we had a whole bunch of enzymes. We had a topoisomerase that opens the origin of replication, another topoisomerase that untangles the DNA, a helicase that unzips the DNA, the single-stranded binding protein preventing the DNA from becoming double-stranded, the primase laying down the RNA primer, the DNA polymerase 3 making the DNA polymerase 1 removing that primer, replacing it with DNA. What's the last enzyme? The last enzyme? I know you didn't study it yet. The last enzyme is DNA ligase. So a whole bunch of enzymes that you need to know by 1 week from today.
25 years of experience.
Yeah, so it's obvious. A little bit, yes. But in transcription, it's only one enzyme. Thank God. RNA polymerase. What you don't know about RNA polymerase is RNA polymerase is actually a collection of enzymes. So the only difference between DNA replication and this one is that DNA replication, these enzymes come by themselves, one at a time, in a specific order. This guy, all the enzymes are coming together as a group collection. So RNA polymerase has helicase activity. RNA polymerase has polymerase activity, of course. So it has all those activities built into it. So we're not going to name each little part of it. We just call it RNA polymerase. The RNA polymerase will bind to the promoter, the light switch, to initiate transcription. But a lot of times, this RNA polymerase has to be invited to bind to the promoter through transcription factors. So these transcription factors in eukaryotes, they actually have to bind to the promoter first. And then they tell RNA polymerase, all right, come on over. We're ready. So anything that brings the RNA polymerase to the promoter is called transcription initiation. Let me tell you something. Everything you do in your life, if you think about it, has three main events: initiation, elongation, termination. Including your life, actually. You're currently, right now, in the elongation state of your life. The initiation was when you were born. And after a long life to you and I, we will eventually reach the final stage, termination. Even coming in here today to take the exam, when you came in at the beginning, that was initiation of the exam. And when you were taking the exam, what was that? Elongation. And then finally you gave it to me. What is that? Termination. You were very happy about that part.
No, not really.
Not really. Everything has those three steps. Why not transcription? And why not translation? So pay attention to this. I'm going to talk about transcription initiation, elongation, termination. And then I'm going to follow through. I don't know if I have time to do all this, but I'll follow through with translation, initiation, elongation, termination. Because everything in my life has those three events. So all the events that bring RNA polymerase to the promoter is transcription initiation. So we're going to play a little game here. Watch us. A little demo. Here, this is the promoter, the light switch. So this is gene expression is off. When the light is off, gene expression is off. I am RNA polymerase. I woke up very early today. I drove all the way over here. I gave you guys the exam. We took the exam and I started talking about things and now here I am. Look, look, look. I'm walking to the promoter. As soon as I touch the promoter, everything that happened today in the morning until this point is transcription initiation. Watch elongation. Watch this. As soon as I press the on button, that's elongation. And then eventually, of course, I press the off button. What is that?
Termination.
Termination. So the way it's drawn right here, that's me touching the light switch. That's the RNA polymerase bound to the promoter. That's transcription initiation. Now watch the elongation now. You should see RNA being made. There it is. What's going to happen now is that the polymerase, the RNA polymerase, will slide through the DNA. And as it does it, it goes through the DNA, it breaks the hydrogen bonds, and it starts copying the RNA. Breaks hydrogen bonds, starts copying the RNA. Breaks hydrogen bonds. So now the RNA is getting longer. Notice that after it leaves, the hydrogen bonds reform. So it unwinds the DNA a little bit, copies, and then moves through it. And then the hydrogen bonds reform after the polymerase passes by. And it just keeps going. Keeps going. So all of this is transcription elongation because it's making the RNA. Until it hits something called the terminator sequence. There's going to be a sequence at the end of the gene that tells the RNA polymerase, you've reached the end. Get off, you've reached the end. So this sequence will tell RNA polymerase, you're done, leave. So that is obviously transcription termination. There it is. So by then, what do you have? You have a full-length copy, exact copy of the gene. Introns and exons if it's eukaryotic. And basically this basically happens exactly the same way in bacteria and in eukaryotes. Questions? Okay, now let's look at eukaryotes. Now I have the RNA in eukaryotes. It's the exact copy of the gene. At the end of transcription, what will I have in eukaryotic cells? I'm going to have a pre-mRNA. It's actually not an mRNA yet. It's called a pre-mRNA. So transcription in eukaryotes produces a pre-mRNA, an exact copy of the gene. Introns and exons both included. This is what I would give you and tell you, here, do whatever you want with it, but don't touch the DNA. So what are you going to do with this pre-mRNA? How do you make the pre-mRNA a mature mRNA? This is the one leaving the nucleus. The mature mRNA, not the pre-mRNA. The pre-mRNA will never leave the nucleus, and if it did, you're not going to get catalase, for example. Remember, I told you the introns have to be removed. So if you let this one go out of the nucleus for translation, you're not gonna get catalase. What are you gonna get? I don't know, but it's not catalase. So it's not gonna leave. The pre-mRNA does not leave the nucleus. It can't anyways. Why? Because it has to be processed. It's kind of like your factory. You have a box of chocolate. Well, you're not gonna just ship it out to your customer until you wrap each chocolate, make it nice and neat, put it in nice gold foil or whatever, put a ribbon and look, then you send it to them. Because you want to make it look nice so they can buy it again. So you want to make it look nice, make it functional. This will not give you catalase. No way. You have to process it. So that's called RNA processing. This does not happen in prokaryotes. For most of the genes in prokaryotes, RNA processing does not happen. The first thing that has to happen to your mRNA, your pre-mRNA, is that you have to put a cap on the 5' end of the RNA. See, you have to tell the ribosome which end is which. In the next step, the ribosome has to bind to the 5' end, and only the 5' end. So how is the ribosome gonna know? You put a cap on it. You put a cap here. So when the ribosome comes, it'll go, oh, there it is. And it will bind here and go this way. So that's the first step of RNA processing. It's called 5' capping. It's no different than you taking a cap or a hat and putting on, hopefully on your head, before you walk outside the door. You never put it anywhere else except on your head. So 5' cap goes on the 5' end of the pre-mRNA. You're already processing it at this point. The next step is removal of the introns. And that's called splicing. And you see that here. It's removing the introns. And remember this part. You have to splice together the exons. So it's like a cut and paste. You cut out the introns and you paste the exons. So now you're going to have one strand where all the exons are fused together. And there are no more introns. You'll have the cap. And then the final processing event is adding a stretch of adenines. Adenine, I don't know, a couple of hundred of them. At the 3' end. So that's called poly-A tail addition, because it ends up looking like a tail. But it's all made up of adenines. Now let's pay attention to the importance of these things. The 5' capping is important for ribosome binding. You know, if I remove this cap, and let's say this mRNA leaves the nucleus, it will not get translated. Nope. Because let's say I remove the cap. The ribosome is gonna come over and go, I don't know which end is which, forget it. And it will not attempt it. So the 5' cap is important for ribosome binding. The introns, you have to remove them, because the introns will not have anything to do with, in this example, catalase. So they're called intervening sequences that need to be removed. The poly-A tail, one of the most important functions of the poly-A tail is that it guides the mRNA out of the nucleus. The poly-A tail is the reason why the mRNA leaves the nucleus. So here we go again. If I cut off the poly-A tail, this mRNA will not be translated, but for a different reason. because it won't be able to leave the nucleus and the ribosome is outside. So the 5' cap, I remove this one, it will not be translated because the ribosome cannot bind to it. If I remove the poly A tail, it will not be translated because it will be stuck in the nucleus. Nice question on the next exam. You have to know what the function of these things are. Now I have everything. Now this one will leave the nucleus. You'll have the cap, the splicing is done, yeah? The poly A tail, everything ready, yes, I'm mature. This is the one that leaves the nucleus, and this is the one the ribosome will bind to. Again, this happens in eukaryotes, not most of the prokaryotic genes, no, that does not happen. There are some unique things that we don't need to get into. More advanced course will cover that for you. All right, so we covered transcription. Any questions before I ruin your day even more? Okay, translation. Again, there's translation initiation, translation elongation, and translation termination. In translation initiation, be careful with this. The small subunit of the ribosome is the first thing that comes in and binds to the 5' end. So by then, this guy is capped in eukaryotes. So the small subunit, so a lot of students by mistake, they think both subunits come in at the same time. No, the small subunit comes in first, binds at the 5' end of the mRNA, the mature mRNA. And then look up here. All it's going to do is scan by reading, of course. It will actually read the sequence, but look what it's going to do. It's going to scan looking for the first AUG. So it's subtle, but I need you to listen, watch. I'm going to pretend I'm the small ribosome, scanning for the first AUG. You're going to notice something. I'm going to read it differently in the beginning compared to when I find the AUG. Watch this. I will read this as C, G, G, A, C, U, C, G, G, A, U, A, AUG, CGC, AAA. Did you catch the subtle difference? In the beginning, I was reading one nucleotide at a time. But once you get to the AUG, the first AUG, you read them as a group of 3. Does anyone know what that's called? It's a codon. Yeah. You have to identify the first AUG. From that point on, it's going to read them in groups of 3s. And these are called codons. All right, so the small ribosome binding to the 5' end and scanning for the AUG, that's all part of translation initiation. And then, watch this. I found AUG, yeah. The first tRNA will now be invited in, along with the large subunit. This together, you have not elongated it yet. You just assembled everything, getting ready for elongation. That is called the translation initiation complex. It's all the stuff that you have to put together, yeah, to start translation. You haven't translated anything yet. You just assembled things. So let's see what's assembled. You have the small subunit at the AUG site. That's called the start codon. And when the large subunit comes in, first of all, the initiator tRNA, which is the first tRNA that comes in, is going to come in and bind to the AUG, as you see here. And the large subunit that comes in, pay attention, it has 3 chambers, EPA. When it comes in, obviously it has the option of E being here, or P being here, or A being here. But actually it doesn't have an option. It will always be P going here. So when the large subunit comes in, it automatically positions the P site right over the initiator codon. Sorry, the initiator tRNA and the start codon. It will never put the E site here or the A site here because it wouldn't work. So it's always the P site is what sits right over the start codon. That means the E site and the A site will be flanking. The E site will be on one side and the A site will be on the other side. This is the translation initiation complex.
So they'll still be there, but not locked in.
Yeah, well, this is locked in right now through hydrogen bonding between these two. There's going to be hydrogen bonding between the U and the A and the A and the U and the C and the G. Now, why is this U? Because this is transfer RNA. So this is an RNA molecule, and this is an RNA molecule, the mRNA. It's complementary base pairing. In RNA, every C goes with G. Every G goes with C. Every A goes with A goes with A goes with I'm still hearing T. A goes with U. U goes with A. They're both RNA. There's no T. So every A goes with U, every U goes with A, every G goes with C. By the way, they're anti-parallel. Notice that this A, look up here, is on the 5' end. And this G is on the 3' end. But look at this. The U, which is this one, that's the 3' end of this tRNA. And the C, which is this one, that's the 5' end of the tRNA. So when you go like this is anti-parallel to this one. It goes opposite each other. That's very important to know. You will never have 5' facing 5'. Never. And you will never have 3' facing 3'. Even here. Now a lot of people don't write 5', 3', 3', 5', but we do write it here to stress that point. Again, another awesome question on an exam. You got to look at the pictures when you're studying. Okay, so this is the initiation complex, and the next event will begin elongation. What's going to happen next is the next tRNA is going to fly in, and you'll see is going to enter the A site. And which tRNA will that be? The one that's complementary to CGC. So what would be the complementary sequence to CGC? Every C goes with
It's gonna be
G. Every G goes with
GCG.
Yeah, so GCG. Here's the amazing thing. How does it know which amino acid to bring in? That's, I was really puzzled when I was a student about that. The idea is this. There are a whole bunch of tRNAs in your cytoplasm. The ones that have UAC as the anticodon, see, this is the codon, this is the anticodon. The anticodon is on the tRNA. Out of all the tRNAs in your cytoplasm, the ones that have the UAC as the anticodon will always be loaded with methionine. That's how it knows. The UAC will always have methionine loaded on it all the time. So it's not like it checks to see what do we need to load. No, it knows it's loaded with methionine. All it has to worry about is the base pair complementarity. The A has to go with U, the U has to go with A, the G has to go with C, and automatically a methionine will be delivered. The next one that comes in play, the same thing. GCG will be the anticodon. And it will deliver an amino acid that's specific to this codon. To know which amino acid comes in, you have to read the codon, not the anticodon. And we'll show you the table for that. So look, with this sequence in hand, I can actually predict the protein sequence. All I have to do is read AUG. Oh, that's going to give me methionine. CGC, I can't remember all that, but we'll look it up. AAA, I think that's phenylalanine. UUU, and so forth. So you're going to have to look at the genetic code, the codon table, and you'll be able to predict the sequence of the protein. But look and see, the next tRNA is going to come into the A site. There we go. The next tRNA with the GCG anticodon comes into the A site. And I already have it up here, delivering arginine. And I'll show you how to look that up. It goes into the A site and settles in. And now hydrogen bonds hold these things together. Now watch this. Look up here, please. The next event is this amino acid, which is methionine, will be cleaved off, and it will go and transfer and bind to the arginine. So now you'll have a green and a red, which means this one here will now be empty, meaning it will not have an amino acid anymore. So I wish I did one more here. It would have been nice to see this red one with the green one while this one is empty. Now watch what's going to happen next. This one you have to watch an animation on it. I'm not talking about my YouTube video or lecture, no. You should find an animation on this, and I'm sure I have some in my resources there for you to use. Watch. The ribosome is going to translocate one codon over. If you can imagine this guy here standing still. It's already hydrogen bonding, so the tRNAs are standing still. It's the ribosome that moves over, one codon over. And when that happens, if you can imagine it here, whatever is in the P site prior to the ribosome moving is now going to be in the E site. Can you imagine that? Let's try it here. Here are the two. Here's the one that came in with arginine, and here's the one with methionine, and here's the EPA. EPA. Sounds good? Like that. If I can fit them in like that. Here it is. When the ribosome moves, watch. Like that. So whatever that was in the P site will now be in the E site. And whatever that's in the A site will now be in the P site. Look, whatever is in the P site will now be in the E site, right here, the empty tRNA. Whatever is in the A site, which will have the green and the red, will now be in the P site. And what's going to happen to the A site? It's going to be empty again. Am I right? So everything shifted over. It's the ribosome that's moving. Now why? Why is it called the E site? The E site is for exit. That means this tRNA, we don't need it anymore. It will leave, see? And it will get reloaded with methionine in case you need to use it again later. At the same time, what's at the P site? The methionine and the arginine. I'm going to ask a question. What is the bond between the methionine and the arginine? What's holding the methionine and the arginine together?
Hydrogen?
Not hydrogen. Peptide
Oh,
bond.
peptide one.
Peptide bond. They're amino acids.
Amino acids, yeah.
Yeah. The next tRNA is going to come into the A site. The new tRNA that comes in always comes into the A site. But for this guy to come in, remember, you have to empty the A site. How? By translocating the ribosome. Now the A site is empty. Now you have AAA. Looks like, yeah, AAA codes for lysine. So now you have AAA, and here comes your lysine. Using this tRNA, UUU is the anticodon, lysine is coming in. By then, this guy already left, leaving this guy empty. And then the same thing happens again. Watch. These two amino acids are cleaved, and they both go over here. So now you're going to have red, green, purple. And it's going to repeat again, and again, and again, until it gets to a stop codon. Once it gets to the stop codon, that's what the stop will do. It will say, you've reached the end. The ribosome, when the ribosome gets to the stop codon, it's going to be looking for a tRNA that's complementary to the stop codon. It doesn't exist. There's no tRNA that's complementary to the stop codon. So what happens is it stalls. Right before that, this ribosome was moving. It was going right through it. And then it gets to the stop codon, it has to stop looking for a tRNA that does not exist. So something called a release factor, it's a protein. A release factor will bind to the A site, not a tRNA, right over the stop codon, and it will tell the whole thing, what are you waiting for? You've already reached the end, stop, disassemble. So the release factor will result or cause the disassembly of the whole thing. The small ribosome will leave, the large ribosome will leave, the tRNAs are done, and what do you have left? The protein that was just made. The protein will undergo folding, it will know exactly where it needs to go, and it will do its job. Questions on it? You gotta watch an animation on this. Okay, let's look at the genetic code. When you, if I give you a sequence, and I will, and ask you, can you tell me what the protein sequence that will come out of this mRNA? You should be able to do that. All you have to do is look for the first AUG, pretend you're a small ribosome. Look for the first AUG and start working with it. But you have to read the sequence on the mRNA. Don't make a mistake and read anything that has to do with the tRNA. This is the genetic code, but this is using the codons, not the anticodons. So I'm going to give you an mRNA sequence, or I'm going to ask you to produce an mRNA sequence, better yet. I'll give you the DNA sequence, ask you to transcribe it. Yeah, good. You got it? Good. Translate it. Do you have to memorize this?
No.
Absolutely not. There's no way. This you will have on the exam, of course. You just have to know how to use it. There are 64 codons. If you don't believe me, go ahead, count them. There are 64 codons. 3 of them are stop codons. Here they are. If you were taking the MCAT, you would have to remember those. Luckily, you don't have to do that. You will have access to this on the exam. So UAA, UAG, UGA, these are referred to as stop codons. This is what terminates translation. And of course, they're going to be at the end of the mRNA. So what's at the beginning? Near the beginning, the start codon, AUG. Now that one you need to know because you need to search for it, right? So it's not that bad. AUG is the start codon. The first AUG is the start codon. Now here's the difference between bacteria and eukaryotes. In eukaryotes, the first AUG codes for methionine. In prokaryotes, it codes for F-methionine, a modified methionine. So I can give you a protein sequence and ask you, is this eukaryotic or prokaryotic? You're gonna look at me like, how would I know? No, you should think about it. How would you know? Where would you look? You would look at the first amino acid. The eukaryotic protein will have methionine as the first amino acid. The prokaryotic protein will have F-met as the first amino acid. And as soon as you see F-met on a protein, you're like, that's bacterial. Absolutely. So that's another difference. Besides the fact that bacteria, they do co-transcriptional translation. Besides the fact that bacteria do not have introns. Bacteria use operons. Bacteria put F-met as the first amino acid, not methionine. It's F-met. Bacteria uses 70S ribosomes. Remember that?
Yep.
What do we use? Mostly is
ADS.
ADS, unless you're in the mitochondria. That's a different story. So those are nice differences that I can obviously formulate a question around. Out of the 64 codons, 3 of them are stop codons. One of them actually is an interesting one. It serves as a start codon and brings in the first amino acid. So it does both of them. Now, what would an internal AUG do? Now, here's the interesting thing. The internal AUG, like an AUG in the middle of the sequence, it's not a start codon. Why? Because it already started. So an AUG in the middle of the sequence just brings in methionine. That's it. Because translation has already started. So the only AUG that acts as the start codon is the first AUG. Now, how do you read it? Well, each codon has 3 positions. And it's always this way from 5' to 3' this way. So when you're reading the sequence, you're gonna read it 5' to 3'. And let's say you read GUU. G is the first position. So you look on this side and you say, oh, here's G. So GUU is somewhere here. Okay, second position, U. So what you're doing is something called triangulating. You're homing in on the codon. All right, G is somewhere here. Second position, U. U is up here, so GUU is somewhere in this box. And then the third one is U. Oh, here it is. I know. You're sitting there. I can just look at it. Why do I have to do all of that? Yeah, because you don't know, am I going to give you this codon table or not? There are other codon tables. You know what they do? They don't give you these. So you have no idea what I'm going to do on the exam. I'll give you one, but it may not be this one. This is the real nice one. This is the one that allows you to do this, or just simply look for it. But what if I give you one that looks like this? None of these are here. All I write is, whatever this codon is, yeah, it codes for alanine. So what is this codon? It's GCU. See? GCU. Okay, whatever this codon is, yeah, it also codes for alanine. What is this second codon here? Let's see. It's GCC. Oh, there it is. So you get used to doing that.
Yep.
Don't assume that I'm going to give you this one. No, because that could be part of the question. Part of the question could be, can you use the codon table without having access to these? I can, it's easy. First position, second position, third position. So I need you to find, without searching for it like this, I need you to find AAA. What is that code for?
AAC.
Lysine. Good. GCA. Alanine. Hopefully you're not shortcutting it. Always practice this. Now, if I give you this on the exam, you can silently say thank you. You don't have to say it out loud. But if I give you the other one, you should also say thank you.
So.
Why? Because you still know how to do it. Yeah.
No, I'm just saying it out loud, like
Yeah.
first.
So let's try it. You tell me. AAC.
So I'm looking at the first.
First is A. So you're somewhere here. Am I right?
Yeah.
Second position is A. So you triangulate. See that?
Yeah.
Third position is C. Did I say C? Yeah. It's not that bad. With a little bit of practice, you'll be a professional at it. Some characteristics of the genetic code. The genetic code is said to be, listen to it, redundant. Redundant means repetitive. If you come in here with 5 cell phones, I'm going to go up to you and say, when it comes to your cell phones, yeah, you are redundant. Redundant means what? Multiple codons code for the same amino acid. That's what it means. In some cases, in a lot of cases, let's see. Multiple codons code for the same amino acid. Multiple codons code for the same, oh, look at leucine. Oh, 6 of them. But then you have 1 or 2 that are not redundant. What's the different way of making methionine? There's only one way to make methionine. And it is AUG. Do you see methionine anywhere else?
Nope.
No? What's the other one? UGG, tryptophan. Any other way of making tryptophan? No.
Nope.
So redundant means multiple codons code for the same amino acid. But then the genetic code is also referred to as not ambiguous. And this one, I will lose half of you. So pay attention, I know it's late in the day. Not ambiguous means every codon, yeah, codes for 1 amino acid. And only 1. And thank God for that. Why? Because if it didn't, we would not care, because we would not be alive. Who? All of us. No life on earth. Example, you. What does you give you? You. Phenylalanine.
Phenylalanine.
Does you give you anything else?
Nope.
No. Why? Because the genetic code is not ambiguous. Ambiguous implies something like vague. Like if I say to you, what does you give you? You say, well, sometimes phenylalanine, but sometimes lysine. You know what that means? That means sometimes you'll be alive, and other time you'll be dead. I'm more worried about the time that you're dead, not the time that you're alive. Life would not exist. You gives you phenylalanine, and I don't care who you are. If you're a tree, human, bacteria, the genetic code is not ambiguous. Whether the gene is coded for in your heart, your liver, your kidney, you will always code for phenylalanine and only phenylalanine. And what do we call that? Not ambiguous. You will never say ACA sometimes codes for this and sometimes codes for that. Then we wouldn't be able to predict the sequence of the protein. If I give you a sequence of mRNA and ask you, listen to me please, hang on a second. I still got 1.5 minutes with you. If I give you a sequence of mRNA and ask all of you, and there are 22, 23 students here, give me the protein sequence. If the genetic code was ambiguous, I would have 23 different answers. Life cannot exist this way. It doesn't work this way. But because we're going to use this, and this guy is not ambiguous, and now we can have the same exact sequence. We can predict the protein sequence because we will be using a genetic code that is, I know it's redundant, but it's also not ambiguous. Sounds good? And we'll practice all that stuff. I'll see you Tuesday.